Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Leisha Koop, Senior Field Application Scientist for Mirus Bio.
Thanks so much, Alex, and thanks to all who've taken time out of their day to join us for a discussion on our breakthrough AAV enhancer to improve titers and reduce costs with any serotype, suspension, and hex cell type, and transfection reagents. We'll start the presentation with a short background on Mirus Bio, followed by a summary of cell and gene therapy and the key manufacturing hurdles that we're facing now in the field. We'll then introduce our novel AAV enhancer, which was designed to improve titers in any suspension hex transfection system, really regardless of the transfection reagent used, the hex cell subtype used, transfection media, or serotype. And as with any process, there are nuances to optimization that ensure you achieve max titers, develop a scalable workflow, and reduce production costs. So we'll go into a few of those, as well as how Mirus is really a trusted partner for R&D through GMP efforts. And then we'll use the last few minutes to address questions in the Q&A session. One of the most important things I think to note about Mirus is that we've been firmly rooted in science for the last 25 years, and we're really passionate about transfection and aiding however possible in the development of life-saving therapeutics. And this is reflected in our mission statement, which is to deliver innovation, empower discovery, and improve life. And just as we step back a bit to explain, we were founded by 3 researchers out of the University of Wisconsin-Madison, and we're still headquartered there today. But we are a global company with distribution networks and sales support in really all major markets. Our focus back then at the start was in vivo non-viral gene therapy, but of course the utility of our novel lipid and polymer libraries for transfection applications really shifted our focus to support all transfection cell biology applications. And then of course the invention of our virus gen products brought us really full circle and back to gene therapies, though this time by AAV and LV, which of course are viral vectors. But through it all, Mirus has been a unique solutions provider in that we partner with you to simplify optimization, to troubleshoot issues, and to really move your projects forward. So in this first section, we'll give a quick review of the state of cell and gene therapy, the critical problems that we're facing in the field, as well as how Mirus seeks to help scientists overcome them. The potential for cell and gene therapies to improve the human condition is undeniable, and we're starting to see the true power of viral vectors for curing cancer and correcting heritable diseases such as spinal muscular atrophy and hemophilia. However, as demand grows and additional therapeutics are approved by the FDA, also undeniable is the need to make these therapeutics in a much more sustainable, affordable manner. AAV-based therapies represent some of the costliest drugs on the market, with some exceeding $3 million per dose. And this massive price tag was tolerable at the start, and with the first few therapies to hit the market, because the therapies were targeting rare diseases with relatively small viral doses and small patient populations. Often adherent hex cell systems were used, despite the difficulty with scale-up and lower titers, but that was tolerable. Now that the potential is realized and larger doses or larger numbers of doses are needed, we're seeing this shift to suspension cells, which are definitely more scalable, or intensified processes like perfusion or modernized fixed-bed bioreactors, all of which lead to better and more cost-effective processes, but with a price tag still exceeding $3 million, even with some of the more modern processes, there's still a lot of room for improvement, and that starts with improving AAV titers. So needless to say, we need more, and it needs to be both safe and cost-effective for everyone in the field. And this is exactly the motivation behind the suite of transit transfection reagents developed by Mirus scientists, which deliver on superior performance versus PEI-based reagents for really all applications, though of course today we're going to focus specifically on AAV. Our reagents incorporate proprietary cationic polymer and lipid technology, which will both bolster transfection efficiency and avoid that excessive cellular toxicity that we see with so many different formulations, in particular liposomal formulations. And this multifunctional formulation addresses several barriers that often prevent effective transfection by other reagents. So the cationic polymer component assists with barrier 1 by condensing nucleic acids, enabling localization to the cell membrane for endocytosis of the complex into the cell. And the lipid component assists with endosome release, so nucleic acids reach the cytoplasm of the cell. And relevant to today's conversation, of course, Mirus has developed enhancers designed to optimize cellular resources for increased transgene expression and viral vector production. The same motivations applied to the Revit AAV enhancer development process. Candidate enhancers were selected through literature-based searches and encompassed several classes of cellular modulators that we felt could improve viral vector production. The key performance indicators for selecting molecules were that they were at least produced a 1.5-fold increase in genome titers or a 1.5-fold increase in percent full capsids without any negative impact on genome titers. So these figures represent hundreds of iterative testing conditions which span a range of operators, suspension HEC293 cell subtypes, transfection durations, pDNA doses, and complex formation times. And as the graph at left shows, several candidate enhancer conditions achieved at least a 1.5-fold increase in genome titers, and a subset achieved greater than 3-fold increase. And while the majority of candidate enhancers had little effect on the amount of percent full capsids, the graph at right shows that some were able to significantly increase that parameter as well. So we then collaborated with several other partners, so pharmaceutical companies that we're working with, medical research partners, to prototype test our top 2 candidates. And then, of course, the winner was brought forward and became the Revit formulation. The feedback we received from prototype testers was incredibly positive. The Revit AAV enhancer formulation was, as I mentioned, selected from the top 2 prototypes and launched as a commercial product at the end of this most recent July. And this chart summarizes the results that teams reported with Revit, which spanned many serotypes, therapeutic transgenes, suspension hex cell types, and of course transfection reagents as well. So for AAV2, titers increased anywhere from 1.2 to 4x with Revit over reagent-alone transfections. Pharmaceutical company B tested Revit with PI Pro as well and noted a 2-fold improvements versus their PEI alone condition. For AAV5, customers saw anywhere from 1.6 to 3.8x higher titers. For AAV8, titer improvements over reagent alone ranged from 1.5 to 4x. For AAV9, improvements ranged from 1.3 to 3.5x. And for AAV-RH10, titers increased 1.5 to 2-fold over reagent alone. Really, in every case, Revit increased titers, and in at least half of the serotypes, customers noted an increase in percent full capsid or the ability to significantly reduce DNA needs compared to their current go-to workflow. And this is our final product of the Revit AAV enhancer paired with our Transit VirusGen transfection reagents. And as mentioned before, the Revit enhancer was really designed to be effective in all suspension HEC-based AAV workflows with the goal of improving titers and reducing costs for every team making an AAV-based therapeutic, as we'll discuss in more detail in just a little bit. And this means that Revit is sold as a standalone product that can be paired with any reagent. We do, however, tend to see maximum titer increases when Revit is paired with the Transit VirusGen transfection reagent. And the kit containing sufficient Revit enhancer and VirusGen transfection reagent for a 10-liter transfection is depicted here, but we do also have kits for 1-liter transfections. And both VirusGen and Revit are animal origin free, and neither product requires a commercial license for RUO or manufacturing use. Both also come with dedicated support from our transfection experts, meaning that we are a partner as you develop your platforms and throughout your scale up and tech transfer to GMP. So one additional to note is that VirusGen is also an excellent transfection reagent for lentivirus production in both adherent and suspension hex cell types, but of course we'll focus on AAV today. In this next section, we're going to look at the Revit's AAV enhancer workflow so we can frame the data that we're then going to look at with Revit in various reagents, AAV serotypes, and suspension hex cells. Really another Revit development goal to go along with efficacy in all AAV production systems was a simple, scalable workflow, because not all of them are, and it's definitely a pain point for many of our customers. So here we show the recommended transfection and enhancer addition process with VirusGen and Revit, though of course this would apply to other transfection reagents as well. First and foremost is the routine maintenance of cells. We recommend splitting cells the day before transfection so that cells are in log phase growth at the time of transfection. On day 0, cells should be diluted to the target transfection density with fresh media. Sometimes that's just a little bit of media, sometimes it's a significant dilution. So if they happen to be right on target, that's great. Otherwise, adjust as needed. To form transfection complexes, you'll first add your complex formation solution to a tube or bottle, and that could be PBS or it could be basal culture media. And then you add the plasmid DNA, mix that well. The Revit AAV enhancer is then added to the diluted DNA at a 500 to 1500x dilution, meaning that you would add 15 to 45 microliters of Revit per 30 ml culture. You'll then mix this well and then add your transfection reagent last. After a quick but thorough final mixing step, you'll incubate your complexes stationary for 15 to 45 minutes and then add to your cells. So you'll stick the transfected cells back in your shaking incubator, and AAV is typically ready for harvest 48 to 72 hours post-transfection. So now let's actually look at the data. In this study, we're looking at expression of AAV2, AAV8, and AAV9 following transfection of viral production 2.0 cells or VP 2.0 cells from Thermo Fisher with the transit VirusGen transfection reagents compared to competing reagents, which we will refer to as polymer F and polymer P. Cells were at 3 million cells per mil at the time of transfection, and manufacturer recommendations were followed for parameters such as total DNA and reagent to DNA ratios. In conditions noted with a plus sign, Revit AAV enhancer was added to the transfection complex. And the bars represent genome copies per mil, dots represent percent full capsids, which were determined from the ratio of genome and capsid titers. The take-home message is this. In all cases, the Revit AAV enhancer about doubles titers regardless of transfection reagent used. So if we look at the data a bit closer, a couple other trends emerge. First, overall highest titers are typically obtained with transit VirusGen plus Revit. Other noteworthy observations is that percent full capsids are typically lowest with polymer F. But of course, with both VirusGen and polymer P, we see a slight improvement in percent full capsids when incorporating the Revit enhancer. So definitely a very positive thing there. In these experiments, we really wanted to look at efficacy of the Revit AAV enhancer in various suspension hex cells and media formulations because, as we all know, not all suspension hex cells are created equal. They perform very differently, behave very differently, and therefore enhance differently. So at far left, the VP 2.0 cells in viral production media, or XB293F cells in XB media, were transfected with VirusGen with or without the Revit AAV enhancer. And in both cases, Revit increased AAV8 titers at least twofold, with slight increases in percent full capsids as well. On the right, a proprietary suspension hex cell line that was grown in viral production media as well also shows really nice titer improvements and percent full capsid boosts when transfecting with VirusGen plus the Revit enhancer. So interestingly, this particular cell line, the one at right, did not respond favorably to other enhancer formulations that we had screened. So seeing the positive Revit result in this line was a huge win for our development team and another nod to Revit being universally beneficial for all suspension hex AAV platforms. In this study, we transfected again the VP 2.0 cells growing in either viral production medium from Thermo or the balanced CD HEC293 media from Irvine Scientific to express AAV8 and 9. We used two micrograms per mil of total DNA and a 3:2 reagent to DNA ratio, so that's a volume to weight ratio for all conditions. The gray scale bars are VirusGen alone, and the red bars are VirusGen plus Revit transfections. And results indicate that similar titers and percent full capsids are achieved with both media formulations. And this is really an important finding from a cost perspective, as the balanced CD media is really much less expensive, and also demonstrates that you have this flexibility with Revit and VirusGen, that you're not locked into a specific system. You can mix and match cells and media and still get very promising, very strong results. In this next section, we'll take a closer look at how Revit can be used at scale and reduce the cost burden of gene therapy development and commercialization. GMP-grade plasmid DNA makes up a significant portion of upstream material costs for AAV manufacturing. And with VirusGen, we typically recommend using about 2 micrograms per mil or per 1 mil of culture. And we don't tend to see a benefit of increasing DNA, even if transfecting at densities above the 3 million cells per mil, which is kind of our norm. And that's great, but what if you could use even less? In this study, we explored the impact of Revit AAV Enhancer on AAV2 and AAV5 when transfecting cells with a total DNA amount ranging from 3 micrograms per mil of culture down to 0.75 micrograms per mil of culture. As with data shown before, bars represent GC per ml titers and dots represent percent full capsids. With AAV2, titers decreased by half with reagent alone transfections when reducing from 2 micrograms per mil to 1 microgram per mil. However, the titer drop is significantly less when using Revit. It's really only about a 20% drop, and you do see a really nice increase in percent full capsids. And for AAV5, the Revit AAV Enhancer allowed for a decrease in pDNA doses to as low as 0.75 micrograms per mil, while still maintaining really high genomic titers. And as we see kind of on repeat and with customers, decreasing pDNA doses does lead to that higher percent full capsids almost universally. And that really demonstrates that cost savings and higher quality AAV are achieved with less DNA in conjunction with Revit, which is precisely the finding that we're hoping that all of those that test this product see as well. This next study demonstrates consistent high productivity with the Revit AAV Enhancer for AAV8 in both shake flasks and bioreactors. And for this, viral production 2.0 cells in balanced CD HEC293 media were seeded in bioreactors at 0.5 million cells per mil 3 days prior to transfection. Cells were diluted to 3 million cells per mil at the time of transfection. The shake flask controls were set up per our standard process, which is essentially passaging the cells the day prior to transfection to about 1.5 E6 and ensuring a final density of 3 E6 at the time of transfection by diluting with a small amount of fresh media, as cells will typically more than double every 24 hours. And in both flasks and bioreactors, we verify that the cells are doubling normally and at least 95%, preferably up to 98% viable at the time of transfection. We tested both 1 microgram per mil and 2 microgram per mil total DNA conditions at the appropriate reagent to DNA ratios with the Transit VirusGen reagent plus Revit Enhancer. And transfection complexes for the reactors were pumped in, and for the shake flasks, they were, of course, just pipetted in. And as with AAV2 and AAV5, we see highest overall titers with 2 micrograms per mil total DNA. However, titers at 1 microgram per mil are only marginally lower, and percent full capsids are much improved compared to the control condition, which was reagent alone transfection using the 2 micrograms per mil total DNA. So what we're seeing here is the Revit process is very scalable from shake flasks to bioreactors. We can definitely expect the same results, which is great as we're looking to scale them into even larger. Complex formation time becomes a significant consideration at that larger scale, particularly when multiple liters of transfection complex need to reach the cells in a very finite amount of time. So in this study, we looked at GC per ml titers versus complex formation time and concentration when transfecting VP 2.0 cells using 2 micrograms per mil total DNA and a 3:2 reagent to DNA ratio. The lines with triangle points depict complexes formed at 10% of the total culture volume in viral production media, and the lines with circles depict 10% complexes formed in PBS without calcium or magnesium. The black lines show virus gen alone titers. Red lines depict the virus gen plus Revit titers at each time point. And we find that incorporating the Revit AAV enhancer into the transfection complex has absolutely no impact on complex formation time, and that our complex formation window is quite stable in the 15 to 60 minute range, meaning that we very much expect to see similar titers if adding complexes to the cells at any time point, 15, 30, 45, or 60 minutes within that time frame. I will note that complexes formed at 5% of the total culture volume, so at a higher concentration, they do grow much faster, which means that your complex formation window is shorter because transfection particles, their growth is driven by concentration of the reagent DNA in the mixture. But as we learned on the previous slide, Revit may enable the use of less DNA, which would reduce the concentration of your complex and growth rate of transfection complex particles, thus extending your complex formation window really back to what we're seeing right here. So ultimately, complex formation time should be empirically determined for each expression construct, but our hope is that Revit makes this parameter much less of a pain point. Delivery of transfection complexes is another parameter that we've explored rather extensively to ensure that your complexes hit the cells within your optimal complex formation window. So for very large complexes, say a 20-liter complex for a 200-liter reactor, or even a 50-liter complex for a 500-liter reactor or larger than that, this process can take a lot of time. So in this study, we looked at a range of pump speeds from 1 liter per minute to 3 liters per minute to see if complexes and the resulting AAV titers were negatively impacted in any way by the increase in pressure and shear forces at those higher pump speeds. So we first looked at the complex radius by DLS, which is shown here, and we do see a slight upward trend in particle size from faster pump speeds. But when we look at the true activity of the complexes, we observe very similar titers and percent full capsids at all speeds, which is great to see. So this means that a 50-liter complex can be delivered to cells in under 20 minutes, which does keep you well within the established complex formation window, at least with Transit Virus Gen. We've worked with many teams at 200-liter scale at this point with the Transit Virus Gen reagents, and large-scale complex formation and delivery to the bioreactor is always really a key part of that conversation, of that discussion. And this process depicts formation of a 10-liter transfection complex for transfection of a 200-liter bioreactor, and it really does align perfectly with the smaller scale recommendations that we went through earlier. So essentially, you first dilute your DNA in complex formation solution in, say, a WAVE or a 2D bioprocessing bag, followed by the addition of the Revit enhancer, and then you'll rock to mix it. You then gently add the Virus Gen reagent to the diluted DNA in Revit. You'll initiate that final mixing by rocking at a greater angle for about 5 seconds. You'll stop rocking for the rest of your complex formation window, and then you can pump or drain the transfection and enhancer mixture into your reactor to transfect your cells. So in summary, the Transit Virus Gen transfection reagent and Revit AAV enhancer were designed to meet a need in the gene therapy field by improving transient transfection and optimizing cellular resources for increased virus production. The Revit AAV enhancer offers better yield with any transfection reagent, as we saw. It's incredibly simple to optimize and leads to cost savings through higher titers and lower DNA requirements. And you get a dedicated support team to assist with optimization and scale-up to accelerate your journey from R&D to GMP. So if your team is interested in evaluating VirusGen and or the Revit enhancer, we'd very much appreciate the opportunity to talk with you. And of course, here's our contact information for both myself as well as our absolutely wonderful technical support team. So thanks again for joining this webinar with us, and I look forward to your questions in the Q&A session.
Okay, great. Thank you, Alicia. And as a reminder to our audience, you can go ahead and type in your questions for Alicia now. So our first question is, when will the GMP version of the Revit enhancer be available?
Thanks, Alex. So we will
launch a GMP version of the Revit reagents in 2024. We don't have an exact time frame outlined yet, but what we're hearing is the summer months. So we're really hoping end of Q2, early Q3 of 2024, we will have the fully GMP version of Revit available. And of course, along with that, we will have a residual reagent assay, which any team using this in a GMP setting would need, to verify that there is no residual, compound left in their final drug product. So we'll have GMP and a residual assay ready for you, hopefully mid-2024.
And our next question is, can Revit be used with other brand transfection reagents?
Yeah, and in fact, we have done testing with Revit and every other major GMP player transfection reagent on the market from various other vendors, other sources. And what we see is that it is indeed universal with each one of those reagents, even those that come in, say, a full transfection kit that have additional feeds and enhancers built into their system. Addition of Revit on top of all of that still does have a really nice, at least twofold improvement in titers with even that system. So we've done quite a bit of testing, and for teams that want to see more of that data, we definitely welcome those conversations and could set up calls with their R&D or PD groups afterwards. But yeah, we've tested it all, and Revit works great with pretty much everything out there.
And you mentioned that you don't see higher titers using cell densities higher than 3 million cells per mil, but have you tested in a perfusion system at all?
So at MIRS, we have not, but we have partnered with Repligen, and they, of course, offer tangential flow systems, perfusion-based systems that are intensified such that you transfect at a much higher cell density, so 9 to 10 million cells per mil, and then the system is designed to do a full media exchange every day. And in those systems, they did utilize the VirusGen reagent in our 1.0 kit, and they had tested a bunch of reagents, and it was the only one that gave this multifold increase in titers. And so we know that the VirusGen reagent is great for that type of situation. We understand how complexes need to be formed to get them effectively to cells at such a high density using more DNA. So we know it does work. We don't have the internal data, but we've seen a fair amount, and it looks very, very promising, at least with the Transit VirusGen reagents. We haven't done that testing yet with Revit, but hopefully, the same holds true there too.
All right, and our next question is, have you developed an assay to quantify the amount of VirusGen and enhancer and AAV preps following purification?
We are in process of developing exactly that assay. So that would be what I'd referred to before as a residual reagent assay. And we will have that ready again at about the same time that we have the GMP grade version of Revit ready. For the VirusGen reagent part of that kit, we do have a GMP version of the reagent, and of course we do have a residual reagent assay for the transfection reagent that would that can detect both the lipid and the polymer components of the reagent itself. So yes, already for VirusGen, and to come as soon as possible for the Revit enhancer.
All right, well, it looks like we're running a bit short on time, so if we don't get to your question, we'll be passing them along to Leisha, who can follow up with you directly. So please go ahead and type in any questions you have, and we'll get those passed along. The last question for the webcast is, what other media have you tested for suspension AAV production, and did any stand out as better than others?
And that's a great question, and something that we hear pretty frequently with newer PD teams that look at the number of medias that are offered, and they grow every day. Different companies are offering different media panels. It's a lot to try to digest, and we have tested at least a dozen different medias, and as a rule, they all work pretty well as long as your cells adapt well to them. There's a couple that have stood out to us as performing slightly better than others in terms of just ease of use, the tighter productivity that we're seeing. So our favorites kind of tend to be the balanced CD Hep 293 media from Irvine, and we've seen really, really nice results also with Sativa's peak expression media. Those are both really, really great contenders. And of course, the thermal suite of media is also really good, but the two I just mentioned, so Balance CD and peak expression, at least in our testing, tend to do a little bit better. So definitely worth looking into if you're planning on exploring media.
All right, great. Well, thank you so much, Leisha.
And thank you, Alex. And thanks to everyone for joining.
Yeah. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with the direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.